Hello, BIO178. Welcome to our Week 7 lab. In lecture this week, we talked about local adaptation and how in one type of experimental approach to determine whether local adaptation is indeed happening in populations, you can do either common garden or reciprocal transplant experiments where you are exposing individuals to some kind of environmental stressor, either in the lab or in the field. And then what you're measuring in those experiments is some kind of phenotypic difference, or we call them response variables. Oftentimes, the response variable or phenotype that you're measuring in these types of experiments is survival, whether individuals were able to survive or not, or alternatively, how long they were able to survive. So what we're going to do in this lab is do a type of survival analysis, which is something you could do, for instance, to determine if the survival of two populations, individuals from two different populations, was significantly different following a common garden type experiment or reciprocal transplant type experiment. The particular data set that we're working with this week is another data set from my lab, but from a different project. So previously we looked at some data from the intertidal snail that I work with. We also work on red abalone in my lab. And this project is at a little bit earlier of a stage, and I talk about this in the background video, which is the next video in line if you're watching these in sequence. But overall, when I'm doing these type of molecular ecology approaches, oftentimes the first thing I'm doing is figuring out if there are phenotype differences between different populations. And with the intertidal snail work I've done, I've been working with that system for years now. And so I'm at a further along stage, meaning I have looked at the molecular mechanisms and I know a little bit more about the molecular mechanisms causing those differences. But when I first started that project, the very first thing I did was just look and see if there were differences in survival after I exposed individuals from Northern and Southern California to the same type of heat stress. And so that's roughly where we are right now with the abalone project. Is we're relatively new with this project, so the first thing we want to do is see if there are phenotype differences, particularly in terms of survival, when you expose red abalone, juvenile red abalone, from different farms in California. So there are wild red abalone along our coast, but they're not doing very well right now, and so it's hard to get wild samples. And so for a lot of the red abalone research that's done nowadays, you're actually getting individuals from different aquaculture farms that grow these abalone for a food source to sell to restaurants, for instance, or people like me who want to study them. So that's what we're working with this particular data set, is comparing individuals from two different abalone farms in California to see if they have significantly different survival following exposure to a heat stress mimicking what they would experience out in the wild in the ocean. So that's what we're focusing on for this particular week, is the survival level of analysis, the phenotype level of analysis. And then in future labs and future weeks, we'll look at the molecular side of things. So first we'll focus here on the phenotypes. As always, if you have issues or questions, especially related to installing the R packages we're using this week or not being able to get certain code to work, please let me know. I do not want you spending hours and hours and hours just trying to figure out how to install these packages. So if you run into issues, please let me know sooner rather than later so we can all maintain our sanity and you can focus your effort where I really want you to focus your effort, and not on R install troubleshooting, that kind of thing. So let me know what questions or issues you have. And I hope you're all doing well, and I will see you in the next video.